Okay, so we'll get straight to the introduction. Tonight we'll hear a talk from Dr. Caitlin Parmalee, who's an assistant professor here in the Department of Mathematics. Dr. Parmalee is a pioneer of her chosen field. She has a BA, an MA, and a PhD in mathematics. Something that actually surprised me when I looked. And the last of those, the PhD, is from the University of Nebraska at Lincoln. She's been teaching at Keene State since 2016. 5 years now. Her classes include differential equations, applied statistics, linear algebra, and mathematical modeling and computing, among other things. She's a teaching star and likes to bring her students to conferences in her field where she presents on topics like tonight. Her research areas center around the use of mathematics to model biological and physiological functions, something that you're going to hear about. Her talk tonight is titled Using Network Structures to Predict Activity in Neural Networks. And with that, I'm just going to say, let's welcome.
Thanks, and thanks all for coming. I saw a lot of my students on the Zoom, so hello. So I'm going to talk just a little bit about this project that I've been working on for a long time now. So my original PhD thesis was thinking about modeling a very specific neuron in your eye, not my eye, but salamander retina. And at the very end of my time in graduate school, my advisor handed me like a silly little cute problem where she had this graph and this dynamics, like, can you predict like what this behavior is from the structure of this graph? And it became this project that's now lasted over 5 years. So I want to tell you a little bit about this. So I would be remiss if I did not. If I did not explain to you what a neuron is and what it does. So neurons are the cells in your nervous system. So lots of cells in your brain, they control motor functions, lots of other things. And so sort of biologically, you have the neuron here. It receives inputs at the dendrites. So those are the little spiny things that stick out. And if it receives enough input and reaches a certain threshold, then it sends a signal down the axon here and onto other neurons. Okay, so the synapse, we've got a blown up view here. You've got inputs coming down the synapse to the cell terminal, and it's going to cause these little bubbles. These are vesicles full of neurotransmitters to open up into the synapse, the space between the two cells, bind to the next cell and send the message along. Okay, so inputs are coming in at the dendrites and going out the axon. And that's how the neurons are constructed. And so one of the big questions in neuroscience right now is thinking about connectomes. So these are basically wiring diagrams of various organisms. So the very first complete connectome that we, as humans, were able to complete was the C. elegans, which is like a tiny nematode. It has 302 neurons and like 7,000 synapses. So in 1982, we were able to map the whole thing. And even then, looking at how all of the neurons are connected, there's still tons of questions about like, what is the neuron? What are they doing? Right? How are they connected to different functions within an organism? And then since then, because we can now measure more and more brain activity, more and more neurons at once, we've very ambitiously started thinking about the human connectome. So your brain has 86 billion neurons and like 100 trillion synapses. And so this seems like sort of an impossible task. But there's lots of sort of like research groups or big organizations thinking about this. So one is the Human Connectome Project. So lots of neuroscientists, if they're ever making recordings in the brain, putting it in sort of this open source space so other people can look at that data. And then also, there's a couple that there's one in Europe, one in the US, so one's like Blue Brain, and like the other one's Big Brain or something. And so there's a couple of different efforts along these lines. And also, you can too. So one of the things that came out, this was like 2013, I think, they gamified mapping neuronal connections. So this is out of the lab of Sebastian Sung at Princeton, so very famous neuroscientist. You can sign up, I did, and I can't spend enough time on this, so I don't, but you go through little tutorials and it shows you how to map like where neurons are connecting. They show you this like 3-dimensional scan of like 1 micron of neuronal tissue. And so what they're thinking about is they have these AI technologies that will actually do some of the mapping ahead of time, but there's no, there's still like a 10% error rate. And when you have that many neurons, that's still a pretty big error. And so they need sort of humans to sort of back up this data. So you can too. Okay, so one of the things that I'm most interested in is sequences in the brain. So for example, if I was thinking about place cells, so place cells are neurons in your brain that are coded for a certain spatial location. Okay, so these traces here are from experiments with rats. So the black trace is where they moved in the cage. Okay, and then for each of these 3 neurons, the red dots are where a specific neuron fired. So this top one is neuron 1, and wherever the rat was when the neuron fired, we put a little red dot there. Okay. And so for this one, it's sort of the upper right corner. For this one, bottom left, and this one sort of top left. And so these neurons are sort of coded for that specific location. Okay, so when the animal is in that location, they fire, and when it moves out of that location, they stop firing. And now there's some error. You can see some sort of spurious firings down here where it's not in that area, but brains make mistakes, just like we all do. Okay, but this was sort of like a big discovery in neuroscience, won the Nobel Prize. There's even, if you saw Nora Young's talk, I think 2 years ago now, she talked about grid cells, which are like the 3-dimensional version of place cells. They did experiments in bats instead, where they fly. So really big deal. So here's where the sequences come in. So if you had an animal in like a maze, and this is just a cartoon, it's a very realistic looking mouse here. You're recording from the pink, blue, yellow, and green neuron. So say those circles are their spatial fields that they're coded to. So when I'm at the end of the maze here, the pink neuron's firing. So if I'm looking at a plot over time, at first the pink neuron's firing, then pink and blue kind of fire together, and then blue and yellow kind of fire together, and then yellow and green, and sort of you see this sequence of neurons firing. So the pink, blue, yellow, green sequence. And the cool thing about this, so they do these experiments, they record the animal's brain activity, and then when they like leave the maze, they also record like while they're sleeping and while they're running on a wheel. You see those sequences played back in the brain over and over again, usually at a different time scale. So like they're processing that memory and coding that information. So that's one of the reasons that neural sequences would be interesting to study. And so that's one of the sort of motivations for part of this project. Okay, any questions so far? Okay, so my PhD advisor is a big proponent of beautiful models. So here's what we mean by that. You want a model that is complex enough that we can take care of all of the biological, realistic type things that we want to model, but that it's still something I can do math with. Okay? If you make your model too complicated, then you're not really going to get much out of, like you might get some good predictions perhaps, but you're not necessarily going to sort of understand how that structure is giving that activity. Because if your model's too complicated, you can't see that connection. So in this particular project that I'm working on, we're interested in how are the connections in a neural network going to tell me something about what the network is going to do. So we wanted something where the only difference between two models is just the connections, right? So all the incoming inputs are going to be the same. I want to keep everything else the same except for which neurons are connected to which other neurons. And then one of the things that we're definitely interested is in the different behaviors that you can see in these, I'll show you some pictures in just a second, in these systems. And it's important that I see sort of all of the possible behaviors, right? Because if my model is leaving one out, then I'm not going to get a full picture of what's going on. I can't predict that behavior if my model is incapable of producing it, right? So we're interested in things like stable fixed points, limit cycles, quasi-periodicity, chaos. If you don't know what any of those things mean, and that's okay, and I'll show you some pictures. So I have one equation for the whole talk. Don't run. So this is what the actual model that we use looks like. So it's a system of differential equations. So for every single neuron in my system, say I have n of them, I have an equation that tells me what is the rate of change of the firing rate of my neuron. Okay? And so there's a couple things that go into controlling that. So one of the things is this term here, this is called a leak term. So notice that this is xi, that's the firing rate of my ith neuron. Okay, so just say x1, that's the firing rate of my first neuron. This term here says that if I keep firing, over time I'm going to get tired and I'm going to decrease down to 0. Or the limit goes to 0. Okay, so that's called my leak term. Your neuron can't fire forever without any inputs. And then these terms, so these ones up here, those are capturing all of the inputs from other neurons in your system. So the Wij's, those are going to be sort of the connections between two neurons. So you can actually, in a broader model, set the strengths at different levels. So in your brain is always changing the different synapses, how strong they are, how weak they are, making new synapses. And so in a wider model, I would have a lot more freedom with my my strengths of those connections. But here, because I care about structure, I'm going to have two possible values for W. One, if there's a connection there, and one, if there's not. Okay? So I'm really boiling it down to just thinking about structure. Okay, so I add up all of the firing rates of the other neurons, scaling it by how strong that connection is. And then this data is the outside inputs. So I'm going to let that be constant for everything, because I don't want that to mess up like how the system's going to behave. I just want to know the internal structure, how that's affecting the behavior. And then this weird like brackets with a plus means that if the thing inside this set of brackets is negative, I'm going to set it to 0. Okay, I don't have like a negative input into that neuron. I'm just not getting any. Okay, so equation done. Okay, well, there's one more matrix. Don't panic. So this is like a tiny example. So say I have this neural network. There's 3 neurons. 1 talks to 2, 2 talks to 3, 3 talks to 1. Okay, so I can like code that information in my strength matrix. So I'm actually going to use just these 2 values. Okay, so I could talk all day about sort of like why we choose these types of things. But I'm going to put a -1 minus or plus epsilon if there's an edge there. So I have an edge from 1 to 2. So this entry has a, hey, there's an edge here number, and then a -1 minus delta if there isn't an edge there. Okay. So that's how I sort of code the information. And then MATLAB solves the system of equations for me. And what it does, what it spits out, what I mean by activity or the output of the network, is a firing rate plot that looks like this. Okay. So each of these neurons has a different color trace. So over time, what is each neuron doing? So the blue neuron starts near 0, increases its firing rate, and then comes back down. So the faster a neuron is firing, the more likely it is to be sending information to other neurons. So thinking of this as, am I passing information along? Okay. And so you'll see this neural sequence. So it fires 1, 2, 3. So in the order of the arrows, which seems really obvious when you're like, oh yeah, of course it does. But within the model, we didn't even know that for sure based on how it was structured. So that's great. It follows the arrows, at least in this small example. Okay. So I want to show you a couple of the different types of activity you can see in these networks. So here's a much larger network, so it has a lot more interesting behaviors. So one thing you can get is what's called stable fixed points. So when I say fixed points, I mean equilibrium, right? So if the neuron sort of hits that, like these neurons are all on, these neurons are off, they stay that way. So you see that like same behavior across the whole time scale. So in this example, it's the pink, brown, and green. So it's this like little trio here in the corner. Okay, they're all on, and everything else turns off. Okay, so that's one type of behavior that you can see out of a neural network. These are super well studied, right? In terms of things like memory, we think that these are the types of things that help store memories, right? So this, I'm thinking about the sandwich I ate for lunch, if I stimulate these 3 neurons, it like, I remember it, it was great. And then if I stimulate different neurons, it's going to move to a different memory, that type of thing. That's like an oversimplification, but sort of for this, well enough. So then here's another one. These are what I study, essentially, limit cycles. So this is kind of like you saw with the 3-cycle example, where there was 3 neurons connected in order. Right? So this one is red, orange, and gray. So that's this triangle in the corner. So you can actually see the order here. But then there's some other neurons that are like also turning on in there. And so like one of the things we want to figure out is which other neurons turn on. Why do they fire in that order? Why that group of neurons? But we can have these sort of cyclical behaviors. So this order that you see here, that appears over and over again, and that'll just continue. So this is another equilibrium point. It's just unstable, so it's kind of like wrapping around itself, okay, repeating back and forth. Okay, another one. We can also have chaotic attractors. So this is actually a pretty nice looking chaotic attractor. The only thing that's chaotic about it is that it's trying really hard to be a limit cycle, and yet it keeps like also trying to be a fixed point for half a second, and then it spins back out into a limit cycle. And these pinches that you see occur at random intervals. Right? So if I picked a different set of initial conditions or like ways to start the network moving, it would look slightly different. Okay. So this is the same neural network, and I have 3 different behaviors. Okay, I think it actually has 4 total, but the other one's like another boring fixed point. Okay, so here's the question. How do I know which groups of neuron are going to create attractors? When I say attractors, I mean these behaviors that I see. So a limit cycle is an attractor, a chaotic attractor, a stable fixed point is a kind of attractor, albeit a boring one. So how do we know which groups are going to create one of these attractors? And then if we do, can I predict what order these are going to fire in? So maybe in a 3-cycle, it's not too hard to see like, oh yeah, it'll be 162, because I've got this sort of arrow ordering to follow. But if it was a bigger subgraph of neurons, how do I know what order they're going to fire in? And so one thing I should point out, this network here, producing these 3 behaviors, so I told you that the only difference between 2 networks is like the way that's wired. This is wired the same. How is it giving different different behaviors? So one of the things to keep in mind is that initial conditions in a system of differential equations matters. So it's who do you turn on at the start? So at time 0, which neurons are firing and which neurons are quiet, and sort of how loud are they or how quiet are they? Right? So if I turn on, these 3 neurons to start, it's probably going to fall into this limit cycle. If I turn on the 3 in the corner, it's probably going to hit this stable attractor. All right. So I'll stop and ask, are there any questions? Yes.
What's the measurement of time, the unit of time?
Oh, here, because it's a toy model, there's no unit. It's just saying like, measure this. If we were actually, so we're interested in doing this for like the zebrafish model, there we would actually have a time scale. We'd need to have some more information about, actually, it's on this slide, this is perfect. You can change the time scale by putting a coefficient here. So then we could just scale it by whatever time we're using. So if we wanted to use milliseconds, like that could happen. Yeah, any questions? Other questions? Yeah.
Yeah, so with the attractor concept, that's like the stable organ
Yes.
going to the Milton cycle or something.
Right. Yeah. So it's like it's equilibrium, but they're unstable. So it's kind of like falling in and out of it almost. So you're like repeating something over and over again. It's not just a like straight line. Yep.
Could you repeat the question there, Caitlin?
Oh, she was asking about the limit cycles, if they were also sort of these equilibrium points. And so yes, they're equilibrium points, but they're unstable. So if you're exactly at the equilibrium point, they would look like a fixed point. But if you're even just a little bit off, then they sort of spiral out, and then they have this rotating behavior around that point. Make sense? Any other questions? Yeah.
The wave pattern
Yeah.
that you keep seeing, is that caused by the dendrites, or I'm sorry, the neurons getting tired and then rejuvenating, or is it caused by the neurons not having inputs and therefore not producing firing patterns?
Ooh, so I think it's a little bit of both, but mostly it's whether or not they've received from a neuron that was also firing recently.
Okay.
Right, so if the neuron you're receiving from has fired recently, you're going to get that input. In terms of the getting tired, you're sort of like, once you start the system and it just keeps going, like there's always energy in the system somewhere. You'll see that these go on sort of forever because they're all sending that information back and forth. But I think it's this piece here, we're always getting the same external inputs to all the, so you're always getting something from outside the system that's sort of like revving it up again.
Okay.
Does that sort of make sense? You're getting this constant stream of external input, but it's the same for everyone.
Okay.
And so that what keeps the system going. I think that was the answer to your actual question.
Tiredness is the major.
Yeah, so if I didn't have this, these would die down.
Okay.
Yeah, yep. Yeah, Jim.
One more.
Yeah.
You mentioned your models are focused just on structure. And what I'm wondering, and maybe my question is totally irrelevant, is it okay to not presume that there's different neurotransmitters firing? Are they all dopaminergic neurons? Would it matter at all if you were releasing dopamine versus norepinephrine or something like that?
Yeah, so in this model, we're treating all of the neurons the same. They are globally like inhibitory neurons. So if you had sort of inhibitory reactions, in a realistic model, those the strengths of the synapses would be a lot different. And probably the behavior of the neurons would be different depending on what kind of neuron it is. So that definitely levels the model up to way past currently mathematically tractable.
Right. To your earlier point, right?
Yeah, but like maybe someday, if we can like understand what's going on here, then like those are some next steps that we have to take. We've already even started looking at if you allow for like different weights here. So that's the model I work with. I don't think I ever said it out loud. A combinatorial threshold-linear network. So CTLNs are the models that I use. Threshold linear networks don't have this like only two types of strengths based on connections. You can have lots of different ones, and you can still prove stuff about it, just not as much at this point because there's just so much more variability. It's not as predictable. Not surprising.
Thank you.
Yeah, other questions? Okay, so this is the aforementioned graph that my advisor handed me in grad school when I was tired and writing this paper, and I've been just like slogging through my same results for a long time. She's like, I have this cute little problem for you. So she handed me this graph, and she told me like, this is the order that the neurons fire in. It goes 6, 3, 4, 5, 1, 7. So I was like, okay, 6, 3, 4, 5, 1, 7. So it makes a little cycle in the graph. Why the heck is it that one and not any of the other 5 cycles in the graph, or 6 cycles or 7 cycles, right? Why that particular subset of neurons, and why that particular order? Okay, so this was sort of like the very start of this project, at least for me. So you do what any good mathematician does. You look at smaller examples. Like, I have no idea what's happening with that graph. There's so many edges. I have no idea. So break it down. The very simplest you can get is a 3 cycle. So I'm interested in what's called oriented graphs. So only unidirectional edges. There's no 2-directional edges at first. And I don't want there to be any sinks. So sinks are neurons where they only get inputs. They don't have any outputs. Like the activity just goes there. So I'm not interested in those cases because they produce a lot of the stable fixed points that I'm not as interested in. I want limit cycles because I want the sequences. So for size 3, there's only 1. Size 4, there's I think like 7, but 3 of them are super boring. So here's the interesting ones. In a 4 cycle, it's exactly what you'd expect. It fires in the order of the 4 cycle. For something like this graph here, it's like a 1, 2, 3 cycle, but then there's like this bonus 4 neuron that kind of fires after 3, but it's real small. So I'm like, okay, that kind of makes sense. I've got this 3 cycle, and there's this like little yellow blip that's popping up right after the gray peak. Like, okay. And this one's similar actually. So I got this 3 cycle, and then I have the 3 going to 4, 4 going back to 1. It just has an extra edge that goes back to 2. but it produces the same behavior, right? The same sequence, 1, 2, 3, and then little 4. So the underlines are like little small peaks. And this one, weirdly enough, even in these simplest of examples, this has 2. So depending on what initial conditions you set your system, like which neurons you turn on at the start, it's going to fall into one of these 2 attractors. So there's one that seems to center on the 1, 2, 3 cycle, but then there's also this 2, 3, 4 cycle that's kind of glued together. So I get one of each of those. So they're very symmetric. So like, okay, I can see that. So I'm like, okay, I feel like I understand these a little bit. Let's go a little bit bigger. We'll go a whole 5 neurons. So I'm going to label these based on like sort of what their base graph is. So when I say S, that's the little square graph. And then the other neurons, or sorry, other graphs here, we have like names for them, D, E, and F. So I can add 1 neuron to those and see what happens. All right, so taking that like size 4 graph that I know something about, add 1 neuron to it. How does that change the system? Okay. So like there's like a bazillion pages in the back of my thesis. They're just like pictures of all of these because I'm showing you 4. But any guesses on how many there are on 5? Ballpark it.
120.
Close. It's 152. So on size 5, so I had 7 examples on size 4. I had 1 example in size 3. I had 7 examples in size 4. And now all of a sudden I have 152 examples. I lived with those 152 graphs for like 2 years and just like analyzed the crap out of them. So here's some like sample ones, right? So if I have a subgraph of that F graph that had the 2, 3 cycles, I'm still getting 2 attractors. Like adding this extra neuron, well, now I have like a little red blip here, and this one red isn't even firing, but like the structure is kind of similar, right? I've still got this like 3 cycle of big firing neurons in both of these. So for the 1, 2, 3, and for the 2, 3, 4. And the same thing for the other one. So this one is a 4 cycle with an extra neuron. It looks like a 4 cycle with an extra neuron, right? So I'm starting to see these patterns of structure. So I stared and stared and stared at these graphs. And eventually, I'm leaving out like a whole bunch of banging my head against the wall. I'll spare you. We started looking at things with double edges, which blows up. So there are 16 graphs of size 3. I wrote it down because I knew I'd forget. Okay, so if you have directed graphs, so ones that have the double edges as well, you're allowing for that. There's 16 on size 3. There's 218 on size 4, and there's 9,608 on size 5. Right, so this opened up like a whole new world. So we had to pare it down. So I'm starting real small. I'm looking at a lot of these types of examples, looking at what their behaviors are. And then over time, we started to see that there were certain subgraphs that seemed to matter when it came to looking at the behavior. So things like that 3 cycle shows up all the time. 4 cycles do too. And things like, see if we've got it on here. I don't have one. So a 3 click, which is 3 neurons, all double edges connecting, right? That showed up a lot in the fixed point. Yeah, Grace.
So could you theoretically build a larger?
Yeah, so that's what we're going to try to do. So building larger attractors from these smaller pieces that I know stuff about. So like you start with a 3 cycle, I'm going to add this node in a way that doesn't kill the 3 cycle. Okay, so we're going to talk a lot about survival of these little subgraphs. And so leading right into this. The types of graphs that we kept seeing over and over and over again had a very particular property. So these, we're going to call them core motifs. They're kind of our building blocks of these networks. Okay, so these are all the ones on size 1, 2, 3, and 4. Okay, and the property that they have is there's only one equilibrium point, and it's the equilibrium point that involves all of the neurons. Okay, so there's no like subgraphs that are also equilibrium points. So this has one fixed point, and I know what it is. It's the one that involves all of the neurons. Okay, so these are kind of my basic building blocks. I can't break them down any further. So you've got some classics, 1 point. This is what we call the 2 click. So anything that's all to all connected, double edges everywhere. You've got a 3 click as well and a 4 click. And then my cycles. So I've got a 3 cycle, I've got a 4 cycle, and then some weird stuff, right? These are the most interesting to think about in terms of new stuff that I can build from. So looking at maybe the structure of this one, So the one on the, sorry, I keep bringing back Zoom folks. I'm sorry, Zoom folks. The one that is on the right, it's a size 4 graph on the top. So you have a node 1. That node sends to 2 and 3. 2 and 3 are connected to each other. And then 2 and 3 both send to 4. So the behavior that you get, if you're sort of thinking and guessing, is maybe what you'd expect. So I get, here's my blue fires. You can't quite see, but gray and green actually fire together exactly at the same time. So 2 and 3 fire together, and then 4 fires. So I get 1, 2, 3 together, and then 4. And then this one here is a really similar graph. It has this back edge, which made me think, oh, I'm going to break the symmetry. That's too bad, but it has the same attractor. So something is really strong about these structures of graphs. And then this one's a weird one. This is what we call a fusion attractor. So there is a 3 cycle, 1, 2, 3. This is bottom left, Zoom folks. 1, 2, 3. And then it's all to all connected. So double arrows, everything with 4. So what I get is literally a fusion of the 2 attractors. This one by itself would just be a fixed point, yellow line straight across. This would be my 3 cycle, and that's the behavior I see here. So I get both of them occurring simultaneously, just super weird. Like once you see it, you're like, oh yeah, of course. But when we first saw it, we're like, what is happening? Yeah. I got excited and I talked fast. Are there any questions?
There is a question from the somebody in the chat.
Yeah.
Mel Adams is saying, "Hi, Caitlin.
Hi.
I'm interested in how learning impacts your model. For example, in sports, repeating a movement until we get better at it, so the firing improves. I'm thinking."
Yeah. So that would change. So Mel, that would change in your weights between the neurons. So the weights inside that like W matrix, if you are sort of repeating or learning or using that synapse or that connection a lot, it's going to get stronger and stronger. And so in this current model, I only have 2 weights, and so I don't allow for learning. I'm just looking at like a very short time scale, the neuron or the network as it is right now, not over a long period. What's it going to do? So the threshold linear networks, which allow for varying weights, that would be a little more of what you're asking about, but even those don't change in time. That would be a next level on top of that. So like our weights are always static in this particular model. Hope that answered the question. Any other questions? So here's our proposed rule, right? Every core motif produces an attractor if and only if the fixed point or that equilibrium point that it's corresponding to survives in the full network. Okay, so I want to talk to you about like what does survival mean? But thinking of this giant network here, there's a bunch of core motifs that are hidden in this network. So like 1, 8, that's a 2-click, so that's a core motif. Okay, but it doesn't have a corresponding attractor. Whereas 4, 8, which is here, this one is a 2-click, so that's a core motif, but it does have an attractor. That's this one. It's a stable fixed point. So like, how do I know which of these core motifs is going to make an attractor and which ones aren't? Okay. So here's where the survival comes in. Okay, so I'm going to look at some examples and try to build it up. So this graph here has the 2, 3, 5 cycle. So that's a core motif. It also has the, let's see, 5, 1, 4 cycle. That's a core motif. So it's 2 core motifs. Only one produces an attractor. So only one survives, whatever that means. So like, what's the difference between this 3 cycle and this 3 cycle? That's something we want to like think a little bit about. Whereas in this model, I still have the 2, 3, 5 attractor. I still have the 1, 5, 4 attractor. I also have a 1, 2, 5 attractor, but only 2 of them survive. So like, what is the survival rule for the 3 cycle based on kind of thinking these through? Plus there's this one. This one's one of our favorites. So it's 4 3 cycles kind of glued together in a little pattern here. It creates 4 attractors. They're all chaotic. They're all beautiful. Like, look at these. Okay, but all of those 3 cycles survive, whatever that means. Okay? So maybe spoil it. Okay, so one of the things that previous work that my advisor and one of my collaborators, so Karina Curto and Katie Morrison did, was they actually proved the survival rule for uniform in degree graphs. So when I say uniform in degree, that's every single neuron in your little subgraph has the same number of incoming arrows. Okay, so a 3-cycle, each node has 1 incoming arrow, so that's uniform in degree 1. Okay, and so the survival rule is this subgraph, sigma, survives if there's no node that has more than your in degree, so d plus 1 or more edges, to some node outside. So we call that the target node. So if there's a target node that receives for a 3-cycle 2 or more edges, that's going to kill that core motif. Right? So back to this example. So this one survived, and it's because looking at the outside neurons, this is 1. I don't receive 2 nodes or 2 edges from that 3-cycle. This one, I also don't receive 2 nodes or 2 edges from that 3-cycle. So that one would survive. But this one, 154, which neuron kills 154? 3. Other way, it's a target, so it sends out.
Oh, then 2.
That's for 2. By process of elimination. Yeah, so 2 receives 2 edges from the
Right.
3-cycle. So if you have a target,
Yep.
your 3-cycle points to another node, then that's going to sort of kill that core motif. It's not going to survive in the full network, which means it's not going to produce an attractor. Right? And so for this one, which is the one that survives. So 125 survives. 235 survives. Okay, you have another chance. Which node kills 154?
Now 3.
Now 3, yes. So 3 receives 2 edges from this 3-cycle. So that target node is like this behavior sending to this node is enough to make this not be an attractor. And it's sort of thinking about like, oh, this node is receiving so much that I can't stabilize in just this.
Yes, it's like sucking it up.
Yeah, so like sends it further along. Yeah.
So I noticed in some of your earlier graphs like this, you had bidirectional edges.
Yeah.
Does it work better to model those as 2 separate edges connected to those, or as one bidirectional edge?
I don't think in this case it would matter how you look at it. If it's helpful to draw it that way, it's fine, but it's all about like how many edges are coming in or how many edges are going out. So sometimes when they're bidirectional, I have a hard time counting in and out. So like it might help to draw them that way, but either like whatever is most comfortable for the person looking at the graphs. Okay. So then in this last one with the 4 chaotic attractors, there's no node outside of each of these 3-cycles that receives 2 edges from the 3-cycle. So all of them survive. And we don't know a lot about what makes these chaotic attractors. Like, why aren't they just 4 nice limit cycles like this? Like, we have some sense that if you have like too much overlap between your attractors, then you get this chaotic behavior, but we don't have any way to predict when that's going to happen or sort of how that works. Still open questions. Okay, so this is back to our original one. So I got a bunch of core motifs here, right? And a lot of them don't survive. So like 1-8, this is uniform in degree. How many edges into each node in 1-8?
Oh, 1.
1, yeah. So for bidirectional, for a 2-click, it's uniform in degree 1. So I need to find a node that gets 2 edges from 1 and 8. So what node gets 2 edges from 1 and 8?
9.
A bunch of them, yeah. So 9 does, 4 does. So it's doubly killed, which doesn't matter. So that one doesn't survive. Whereas 4-8, which is here, none of the nodes around it receive edges from both 4 and 8. Okay, so 4-8 would survive and then produce an attractor. And so thinking about sort of the larger subgraphs, so something like, let's look at, 189. So this is uniform in degree 2, because each of those nodes is receiving 2 from the other nodes that are in the subnetwork. And so I would need 3 edges to kill it, 3 edges out. And there's no node where I'm getting from every single thing here. So that one survives and gives. All right, so something large, let's see, 1, 2, 5, is that the one we look at? Yeah, so 1, 2, 5 is like the red, white, and blue here, red, gray, and blue. So looking for, I don't even know which one it is that kills it. I need to find an node outside of 1, 2, 5 that receives at least 2 edges from 1, 2, or 5. 1, 2, and 5.
6.
Yes. So 1, 2, 5, 2 and 5 both go to 6. 6 is the target node that kills 1, 2, 5. So that gets crossed off the list. So I start with like a list of here's all the core motifs that I see in this graph. So this is all of them. And then I go through and sort of systematically, okay, is this going to survive or die based on this uniform in degree rule? So this works for only uniform in degree graphs. You'll notice that there were some core motifs that weren't. So like we still need more rules. Okay. Back to this one. So now, after many years, every single time we came up with a possible way to predict, we would come back to this example and we would try it and everything would break and we would be sad. It's like this example was good to have because everything we sort of hypothesized, like, oh, maybe it's because of these types of structures or because of this type of pattern. And then we'd come back to here and be like, nope, it doesn't work in this example. So it was nice to have this kind of one example that was great at breaking stuff, counterexample for everything. Okay, but now I'm going to use sort of what I've again left out a lot of the inner hard work of like things we tried and didn't work, and made it look really smooth and easy. Start with your full graph, identify any core motifs, and then check if they survive. So this one has some core motifs, only 2 of them survive. So this graph here, this is what we call the 5-star. So node 1 sends to the next 2 in the cycle. 7 sends to the next 2 in the cycle. Each of these sends to the like next 2 around. So it has a nice like cyclic behavior. And then this weirdo over here, which is again, I got a node, it sends to everybody in this 3 cycle. Everybody in this 3 cycle sends to 4, and then it sends back. So like when you look at the behavior, these are going to fire together because of those symmetric inputs. So it's kind of a cool example. So then I look at these and I expect there to be attractors for each of those. Yeah, Grace.
Does your percentage of core motifs that survive like change with depending on how big your network is?
It's not just the size of the network, but also the density of edges. So I've showed you a lot of examples where like this specific example is a complete graph. Well, kind of, because it's unidirectional edges. It has an edge between every single pair of neurons. Whereas if you had a less dense, like there might be less core motifs, but there's also less nodes to like less edges to kill stuff. So like there's this weird balance between what's going to survive when you're thinking about how big is my network and how dense are the edges. So there's sort of lots of things to take into account.
That's why I was wondering about like percentage.
Yeah.
Versus
Like,
actual
who
numbers.
knows? Yeah, it's hard to tell. So my first surviving core motif, that 5-star, I want to try to predict, right? So one thing I know about the 5-star is that these are the order that it fires. So 1, 7, 6, 3, 4. Like give a whole nother talk on why it's not the inner star, which is 1, 6, 4, 7, 3. Pretend you didn't see that, right? So it fires around the outside. And then I'm going to look at how are the other neurons connected, right? So just those core neurons. So the 5 receives from 3 and 4 from the core motif. 2 receives from 3 and 7. So one of the big mysteries about this particular example, like this n=7 example we kept coming back to, is why does neuron 2, this red one, why does it die? When I'm looking at this network here, it has 3 inputs. Like why on earth is that not firing? Especially because 3 only has 2 inputs and it is. So like that was one of the huge things that sort of bothered us about this graph very early on. Like there's more to this than we're realizing. So this kind of gives us a sense. So this is not a hard and fast rule, but at least for the 5-star, it seems like if you receive from consecutive nodes in the cycle, you appear down here. So 5 receives from both 3 and 4, and 3 and 4 fire next to each other. And so 5, it's small, but it's there. Whereas 2 receive from 2 nodes in the cycle that are sort of like 2 apart. Okay, they're not consecutive in that firing order. And that one doesn't survive. So that seems consistent across lots of examples where if you receive from two nodes that are next to each other, you live and appear in the sequence. And so you'll sort of appear, so it's 3, 4. So after 4, it's receiving from both of these. Whereas this one, we don't expect to see because it's not consecutive firing. Questions? Yes.
So would that not fire because like the nerves aren't consecutively stimulating it, so it just doesn't reach that threshold?
I think that's what it is, right? So 7 fires, which like simulates this one, but then it takes a while to get to 3. And so by the time we get to 3, 7 is tired. And so you're not getting enough to hit that threshold. I think you're exactly right. Yeah.
When you say it takes a lot of time, how much time?
Oh, time is meaningless in the model.
Okay.
So like relative to like whatever this mystery time scale is,
Okay.
right? You could, as we were talking about before, you could scale the leak term to create like whatever time scale is biologically reasonable. But here we just chose a uniform coefficient of 1 just to make things easy.
Okay.
Yeah. So time has no meaning. Okay, there was one more surviving core motif and there's one more attractor. So this one, we have the 7 should fire. These I expect to fire together if this was just a core motif on its own, and they do. So if you were to look at the firing rate curves for just this, it would be 7, 3, 2, and 6 would fire at the same time, and then 4, and then it would repeat. But since I've got other things that are feeding in and out of that network, it's breaking the symmetry, and so those aren't necessarily going to fire together, but they should fire like within like sort of after 7, but before 4-ish. So we've got 7 fires, and then I have my blue, red, pink. So these fire together, but orange snuck in there. Orange is one of my, we call them peripheral neurons, the ones that get inputs from that core motif that's surviving. And so this is even, I couldn't even tell you the survival rules for this one. Like I think we have them written down somewhere of like which peripheral, actually we don't for this one. It's like, how do we know which neurons receiving, which neurons you're receiving from allow you to sort of appear in this or not? So with the 5-star, it was like, are they consecutive or not? Here, it's a much weirder sequence, so we don't know this. So I am looking for a student researcher to help me with this core periphery sub-story. So if you're interested, email me. Okay, so then we can kind of see how that sequence is coming from this graph. Like the baseline is that core motif. And then I have this sort of core periphery story where I think I can make some rules about where those are going to appear in the sequence. You can kind of see like 1 is receiving from 4, 2, and 3. So they kind of, the last one is 4-ish. Where does orange appear? Nope, that doesn't make any sense to me. I also can't tell the difference between the orange and the red here. Yeah, it's really hard. I think you're right. I think it's the bigger one here. Yeah, so it's a mystery. Help me, please. Okay. Oh, so one thing that I like kind of swept under the rug when I was showing this example is like, how do we really know the sequence of firing that I'm starting with before I'm adding in these peripheral neurons? So same as this, like it fires in this order, but like there's another 5 cycle in here that it could easily fire in that order as well. I should have brought a picture. So if you change, remember those epsilons and deltas at the beginning? If you change those, then you can actually find missing attractors that like don't appear in certain parameter ranges. So this one, if you just have it in our sort of standard parameters, we always use epsilon 0.25 and delta 0.5. This fires in like the standard 17634. If you are to up the inhibition in your network, then you get the other one, and it's super weird looking. We call it the Gaudi attractors. If you know anything about the artist Gaudi, just Google it and you'll like, oh, like it's like a super weird, it's not these nice like, oh, look at a parabolic kind of curve. It's like very strange. I should have just added it to the end of the talk. I apologize. Okay, so like how do I, from the core motifs, predict that behavior? Like if I know the core motif, I can kind of fill in where the peripheral neurons maybe go. That's something we're working on. But how do I get it from the core motif? So one of the things that we've been working a lot on is these sort of like superstructures in the graph, right? So one of them that we spent a lot of time on is cyclic union. So this is something I noticed pretty early on, that when you have like a node here, and then these three, like it receives from all of them, and then these three point to the next one. So like there's three components, and everything in this component points to the next one. Everything in this component points to the next one. Everything in this component points to the next one, okay? So we call that a cyclic union. So I'm just taking like little subgraphs, these happen to be core motifs, and then I'm like stringing them together and just like adding all directions forward, or all edges forward, right? And so we get sort of a like expected behavior maybe. So 1 fires, these 3 fire together, and then 5 fires. Like, okay, that sort of makes sense. But like not all graphs are going to be nice structurally like this. Like as soon as I add in one edge, I'm breaking some symmetry somewhere, and things are going to happen. So one of the cool things is we sort of started to figure out like what is the generalization of cyclic unions that lets me get away with having edges missing or extra edges backwards, but keeps the structure. So you can see the same type of attractor here. So blue fires, these ones fire like kind of together, and then red fires, same down here, except for it's a little messier. You can still see they're kind of in the same place. Okay, this one, I've got a missing edge here. This one, I have a missing edge and a back edge. Okay, so we sort of came up with this rule, and it has to do with fixed points for knowing whether or not this sort of partition of nodes of this, and this is my 3 components, whether or not that's a directional cycle, so we'll call it. And so it comes down to, if you look at 2 consecutive parts of your partition, if the fixed points all live in the second one. So if the fixed points all live in the second one, you expect the sort of behavior to flow into that one. And so then if they flow into that one, then you look at the next pair. If all the fixed points are living here, it sort of flows into the next one. So directional cycles, like if I were to define them mathematically, are very gross. But that's sort of the idea behind it. So thinking of this, like I'm pulling the flow because I have this fixed point, this equilibrium point that I'm headed towards. And if it's in sort of the second component, that moves the signal forward. So I get this cyclic behavior, even without that cyclic union structure. Any questions? Okay. So here's where we're at. So we can correctly predict attractors for all of those 160. So I said there's 152 on 5. If you add in the 4 and 3 ones as well, that's 160 total graphs. In 0.25.5, so those are the parameters we swept under the rug earlier, right? I can correctly identify attractors for 154 of them. If I modify the parameters a little bit, I can catch all of them. So sometimes there's missing ones in this parameter regime, but if I up delta, I can find the others, right? So they still exist, they just happen to not exist for these particular choices for the weights, because we sort of set those at the beginning. We classified all of those 9,608 graphs based on their surviving fixed points. So that's one of the papers that we're working on pushing out now, is we have these sort of families of graphs that have similar surviving fixed points. So we like reoriented them so they're all kind of like looking almost the same. And you see such similarities in their attractors, like there has to be something to our story, right? Because I said that was a proposed rule. Like y'all just believed me, but like it's not proven. I don't know how to prove it. Like that there's so much like detail and nuance to these systems of equations. Because it's a nonlinear system of differential equations, that's the problem, right? There's not as much like mathematical tool that we have to deal with that. So then the other big part, so this is a paper that we just submitted over the summer, is that directional cycle structure. So thinking about, can I kind of break the graph into these different components and see like where is the behavior going to flow, right? If I can create this sort of cycle, right, then I know, okay, well, these neurons are going to fire first, and then the next ones in the cycle are going to fire. And then if I know something about those smaller pieces, like I kind of break it down. So that's something that we feel like that was like a huge breakthrough, because we understood cyclic unions, but like how to generalize it took a long time to figure out like, okay, like how do I know which edges I can get rid of or which ones I can have a backwards edge as well? That was really hard. And then this is the big next step. So once you see that surviving core motif, you have those peripheral neurons that it points to. Where do they go in the cycle? Do they survive or not? How does that all sort of fit together in the big picture? And so again, plug for a student researcher. If you're interested, let me know. And then, so here's, I want to like definitely recognize Karina Curto, who's my former PhD advisor and still collaborator today, and Katie Morrison, who is the three of us are sort of the core group. And then on this particular project, Juliana Lodano Alvarez is one of Karina's new PhD students. She's been doing some great work on this project, even though she's like in her second year. So it's really fun. And that's it. Thank you. More questions?
I have a question. If you could talk a little bit about like once it is solved, right? And I realize that's a long
Yeah.
ways off. What are, kind of what's the application or like the clinical application?
Yeah,
to the
so
neuroscience field, like how would this be?
so there's a couple of things. So the stable fixed points, like lots of things have been done with that, and that's usually like memory type research and learning. With the limit cycles, that's a lot more maybe like hippocampus, like seeing the rats and their location in the cage, and understanding like how, I guess it's again, it's kind of a storing of memory, but memory of spatial location. What's interesting to me, and I don't know how direct this connection is those chaotic attractors. So like if your brain is producing sort of this chaotic behavior, that's probably not good. Like, is that where like seizures come from? Like, so what structures are causing that type of behavior? So if we could predict some of those chaotic attractors, I think that would be sort of, I think that would be a big win for us, even though like we're so far off from like the realistic application. Although, some folks in France have zebrafish. You want to know why I use zebrafish to look at their brains? They have clear little heads. You can see right through them. So they have this zebrafish data. They actually use a model really similar to ours. And so they wanted to see like, okay, can we look at these structures? We can actually look at the neurons and like how they're connected and see like what the fish is doing at that time. So like that's one of our sort of like baby next steps is baby zebrafish. Yeah. But it's a long time before we get to like solving seizures.
Sure, sure.
Yeah. Other questions?
So I'm trying to picture you as a grad student.
That looked pretty much
Thinking.
the same as now.
You know, engaged in this task.
Yeah.
And you said, you know, staring at it, beating my head against the wall. But you're not just doing that. You're actually looking at the patterns of neurons and then looking at the connections and sort of logicking out which patterns are going to survive and which ones aren't.
Yeah.
So it's a pretty blunt sort of exercise where you're just sort of
Yeah.
putting the pattern down, seeing if it survives, throwing it out if it doesn't.
Well, so at that point, we didn't understand core motifs. We didn't know that there was like these, like why these certain subset of neurons were causing the behaviors. Because in such small graphs, like it was the 3 cycle a lot of the time, and you're like, okay, maybe it's just the 3 cycle that's special. So like in my thesis, I like proposed this algorithm for prediction, which like I was so proud of at the time. We called it the Caitlin algorithm in our group, and it just made me so happy, where you would like take a network and then you would look at who's receiving the least, and you would take that out. And then you would keep doing that until you got to like, okay, well now there's like a 3 cycle, so you can't do it anymore. And then you would reverse the process and like, okay, what's the last thing I deleted? Where can that go in the sequence? And then where can the second to last one go within that whole sequence? And you could like build it back up. And it works for tons of oriented graphs. But since we were limited to the like unidirectional edges, I mean, it doesn't work overall. It was a cool heuristic algorithm, and it led us to core motifs because like, that's what I called them. Like when I got to the end, when my algorithm sort of stopped before I reversed, that was like the core of the graph. It was just a 3 cycle that was left. Now I can't delete anything because they all have the same amount of edges in. So at that point, we didn't really understand the connection between the fixed points and like these core motifs and survival and not survival. Because like MATLAB, can spit out like what all the fixed points are. But we wanted to understand, like graphically, could I tell you what the fixed points are by just looking at the connections? We have a ton of graph rules. So I showed you the uniform in degree one. There's a whole list of different rules that you can decide if a graph isn't uniform in degree. How do you know if it survives or not? Right? Because I didn't talk about that here because that's like a whole nother talk. I gave like 5 other talks.
Core motif is actually a phrase that you and your research group came up with.
Yes.
Okay, and as you took that to conferences and talked about it, people say, yeah, that's a great phrasing. I mean, it's a neat, I just thought it was like the term of the art, you know, and everybody
No.
used it. So is everybody ready to use core motif?
Yeah, I mean, so not to brag, but people are really excited about this particular research. Like when Karina often gives talks at like really big international conferences, she gets invited all the time. And people see this model and they see how good we are at predicting, even just in these small examples. And they're like, ooh, I have a system that looks like that. We can apply it there. But also with the understanding that it's at such a like toy model level where I'm looking at graphs of 5 neurons and there's 86 billion in your brain. So like knowing that we're a little bit further, but like interested in the math, that's hard to do. So like pitching it to neuroscientists, you have to tell a slightly different story than I've told here. Like you would have to rely a lot more heavily on the biological relevance. Whereas I tend to like to talk about the mathy stuff because that's the side that I come from. But people are interested. I don't know. No one's harshed on our lingo. We spend a lot of time thinking about how to name stuff. And my advisor really loves puns. So we've had a lot of rejected ones because Katie won't deal with the puns.
I'm impressed that you're in a position with this research where you can come up with the phrasing that the rest of the rest of the world uses.
Yeah, so Katie and Karina they came up with the combinatorial threshold-linear network. Threshold-linear networks existed, but focusing on these only like there's this weight if it's an edge and this weight if it's not an edge, and studying just that, they were the first ones to do it. And as of right now, we're the only ones doing it. And so we get to name stuff, which is really cool. Yeah. Other questions? Yeah.
You may have said this, but the number of different limit cycles for a particular thing, is that related to the number of core motifs in it, or is it related to something else? And then, can you look at it? Can you look at something and say, oh, I know why, and it's related to different initial conditions, right?
Yeah.
Depending on what you start firing.
Yeah.
Can you predict that? I mean, do you understand looking at the graph why? Oh, if I start with these, this will happen.
Yeah.
If I start with those, this will happen.
It is the survival of those core motifs. So if a core motif survives, we expect there to be an attractor that's focused on those neurons. And then to actually sort of like access that attractor, to like get that one jump started, if you initialize at just those neurons and have everything else off, or like maybe a little bit of noise, then it falls into that attractor pretty standardly. Like there's some where the basins of attraction are butted up against each other, and so it like falls into the wrong one. So you have to like go really small around your fixed point. Yeah, but it's all about the survival of that core motif. If it survives in the network based on our graph rules that we came up with, then we expect to see an attractor, and we kind of know what it's going to look like based on like what the core motif does.
Right.
So that graph, oh, it's a 2-click, so it's going to be a stable fixed point. Or this one was a 3-cycle, so it's going to be a 3-cycle with some like peripheral nodes doing stuff. Did that answer your question?
Yeah,
Okay.
very well.
Other questions?
All right.
Thanks for
Well,
coming.
thank you very much.